Hello friends, whether you are a subscriber or you have just stumbled upon our channel, I am delighted to welcome you to today's exciting video. Let me introduce myself. I am Dr. Kumar and I'm a senior scientist with a PhD in medical microbiology, and I have been conducting research in virology, mentoring junior scientists and research students for over a decade now. And I'm thrilled to have you join us today for a discussion on virus-like particles. So let's get So today we are going to discuss about the virus-like particles, uh, their production, purification, and utilization in research. So what are virus-like particles? As the name suggests, they are particles, they are derived structures composed of one or more different molecules that closely mimic the natural virus in both morphology as well as size. So they are very similar to the natural virus, and they are non-infectious particles because they lack the genetic material. So this is the reason why we call them as virus-like particle and not the natural virus. So this is the natural virus. If you see, the natural virus is composed of the structural proteins, the outer proteins, as well as the genetic material, which you can see inside it. But this is the virus-like particle wherein we develop these particles, or we produce these particles in the lab, and we assemble like these uh, proteins. The structural proteins are same, which is in the natural virus. The only difference is we do not put the genetic material here inside, and that is how this particle becomes safe particle. At the same time, these VLPs retain the immunogenicity. They are a good immunogen. They can be used as a vaccine candidate, and there are a number of uses that we'll be discussing at the end of this video. So the major difference between these two is the lack of genetic material, except that it has all the structural proteins, so it is going to have all the outer proteins that can generate immune response. So let's talk about the history first. So these particles can be either naturally occurring or they can be synthesized in the labs in an expression system. They were first identified in 1968 in sera of patients with the Down syndrome, leukemia, and hepatitis. These VLPs can be formed by either viral capsid protein, the envelope protein, or the core proteins, or a combination of these proteins. So different viruses make VLPs using different, proteins. Either they will need one protein or two protein or three proteins to make the virus-like particles. Now these particles can be produced in expression system, including the prokaryotic cells or yeast system or insect cell lines, even plants or mammalian cell lines. Now these mammalian cell lines have been extensively used to produce these virus-like particles, and the most preferred cell line is the HEK293 cells, which is very happily accepting all those plasmid transfections, and that is the reason that cell line is preferred for making VLPs if you want to make them in bulk. Now these VLPs are generally made for a single virus, but again chimeric VLPs are possible, and they are also made for various research purposes. The difference between these two is the single virus VLPs are made from the structural proteins from one type of virus, while these chimeric VLPs, they can have these proteins from two different viruses, and they both can assemble into one particle, and that is why we call them as a chimeric VLPs. Now that can be studied for various things because it will carry the characteristics of two different viruses, these chimeric VLPs. Now these VLPs have been produced for several viruses till now. We have produced it for the human immunodeficiency virus HIV. We have made it for adeno-associated virus. We have made for hepatitis B virus, hepatitis C virus, bacteriophages, and we have also made it for coronaviruses. Now these coronaviruses have been into picture since several decades, and we have successfully made the VLPs for SARS-CoV-1, MERS coronaviruses, and more lately for the SARS coronavirus 2, which is the reason for the latest coronavirus pandemic. Now let's talk about the production of VLPs, how to produce them in the lab. Now I'm going to discuss some of the basic steps that are needed for producing VLPs in the lab. So you can follow these steps to make your own VLPs in the lab, in your lab. So here it's not possible to discuss the production of different types of VLPs. I'm just going to take one example of a virus-like particle, and I'm going to show the steps that you can use to produce that virus-like particle. So the main reagents and cells needed for these production VLPs are, like, you will need the plasmids. Obviously, these plasmids will bring in all the genes that are necessary to translate proteins which are required to be assembled in the particle, the virus-like particle, and then you will need cells in which you will be doing the transfection and producing these virus-like particles, and then of course you will need a transfection reagent, which, I mean, you can use lipopectamine, or you can use any other transfection reagent, as well as you can use the electroporation if you have the facility. Now again, it's your choice whether you want to go for a transfection reagent or you want to go for electroporation. I have used both, and in my experience, both work well. It's just that it's your decision whether you want to have lipopectamine based transfection or electroporation. Electroporation is very efficient though. So now let's talk about the plasmid construction. Now I'm going to take an example of the SARS coronavirus 2 to show you the subsequent steps how these VLPs can be made. So let's talk about the structure of the genetic structure of the virus. So leave out all other genes. I'm just going to talk about the 4 genes, and or you can say the 4 structural proteins which will be required to make the SARS coronavirus 2 virus-like particles. So for that you require the spike protein, the envelope protein, the matrix, and the nucleocapsid protein. So you need to clone these genes into plasmids, mammalian expression plasmids. You can either clone them, individually in separate plasmids, or you can clone them in one. It's your choice, but both ways, it can be done. So first of all, you need to get the sequence of the genes like spike or envelope or matrix or nucleocapsid. You can get it from the NCBI. So you need to have the sequence of the genes, and then you should consider doing a human codon optimization for efficient production of virus-like particles and further research. And you can clone them in individual plasmids or all in one. As I said, you can use either Gibson assembly or any other traditional method to clone these genes into the individual plasmids. Now you need to do the transfection or electroporation of these plasmids into the choice of cells. Here I am taking a mammalian cell line, preferably HEK293 cells, because as I told, like, these cell lines are very efficiently taking up all the plasmids, and they do not show a lot of adverse response because of the plasmid load. So this cell line is preferred by most of the researchers. So yeah, you can take all these plasmids S, E, M, and N from individual vials, and you can do a co-transfection in the HEK293 cells. Make sure you do the transfection when the cells are around 80% confluence. The reason is you need to allow them to stay in the incubator for another 24 to 48 hours for production of VLPs. So you need to start with around 80% confluence. So by the time it's 24 hours or 48 hours, the cells become 100%, and it doesn't get too much crowded. So what you do is you do the co-transfection of these 4 plasmids in equimolar concentration in the cells here, and you can leave it for 4 to 5 hours at 37 degrees centigrade incubator. And after 4 to 5 hours, you should wash the cells and replace with the fresh medium. This step is important because you need to get rid of the remaining plasmids and the transfectants. So this fresh medium helps the cells to again regain the health quickly. So as I told that after 4 to 5 hours, when you wash them, you need to put them into the 37 degrees Celsius incubator. And now again here it's your choice. People have been collecting the produced VLPs at 48 hours post transfection, or you can do it at 24 hours also. So that is the reason I've given a range from 24 to 48 hours. If you collect it at 24 hours, you will get a fresh VLP, young VLPs, and if you want to collect it at 48 hours, obviously you will get more VLPs compared to 24 hours, but at the same time, those VLPs will be little aged. At the same time, when you transfect cells and leave it for 24 to 48 hours. At 48 hours, there is a possibility that the cells will be showing some adverse reactions, and a lot of non-conventional secretion of proteins or free protein, viral proteins may take place. So you need to be very careful while purifying them. So what I prefer is 24 hours. But again, this is a step where you need to standardize as per your requirements and the type of virus particle you are making. Now, just put them into the incubator, and the assembly and release of the VLPs will happen into the medium here. And at 24 or 48 hours, you just need to collect the supernatant in a tube, and you can now here you can make multiple plates, and you can collect a lot of soup, and you can pool them all in one, or you can start keeping them at 4 degree and do another transfection next day, or third day, fourth day, fifth day, and you can again pool them all at once and then purify them all together. So let's talk about the purification of these VLPs. Now the major reagents that you need for purification is, now again I'm going to tell you a simple way of purification of VLPs. There are other ways also. Here I'm going to explain how you can use sucrose for purifying these VLPs. You will need the DMEM, the media, serum pre-medium, and you will need a refrigerated centrifuge with swing rotors to purify these VLPs. Now, since I'm going to explain sucrose-based, sucrose cushion-based purification here, the other way is to use column-based purification. We can use size exclusion chromatography columns to purify these VLPs also. If you want, I can discuss them, in my next video, if you want them. So let's see how it is done. So first of all, when you collect all the supernatant at 24 or 48 hours, you need to centrifuge them at 4 degrees centigrade. Now from here onwards, keep them at 4 degree or on ice, but try to avoid bringing them at room temperature or higher temperature, because you need to get all the intact virus particles. No damage should be there. So the first step is to centrifuge them at 4 degrees centigrade at 300 G for 10 minutes. This will give you a pellet here. Now you need to transfer this clear supernatant into another tube, and then again centrifuge at 4 degrees centigrade, but with a higher speed of 3000 G for 10 minutes. And again you will see very small pellet here. Transfer this clear supernatant into the next tube, and again spin them at 3000 G for 10 minutes, and you should get a clean supernatant. So you have to repeat this step 3000 G for 10 minutes till the time you don't see any pellet here and it's a clean soup. I'll give you the reason why I'm saying so. You need to get rid of all the cell debris, because when you collect the supernatant at 48 hours, cells also die, and you will get cell debris also in the supernatant. So you need to get rid of all those cell debris. You only need the virus-like particles. So this is the reason this, the whole step is called clarification of supernatant. So you are clarifying and you are getting rid of all the cell debris, and you are collecting a clean supernatant at last in this, clarification step. Now let's talk about the concentration of supernatant now. Now since I told you, previously that you can collect these supernatant from multiple plates and pool them. So if you have a lot of supernatant, then you have to concentrate them before purifying them. So either you can use a 100 kilodalton 15 ml filter, which is commercially available. All you need to do is it comes like a tube, and there is small filter here within the tube. All you need to do is just put in around 5 to 10 ml of your supernatant here, and spin them at 2500 G at 4 degrees centigrade, and you will see that all the supernatant will flow through, and there will be some concentrated soup in the filter here. Again, now here you need to keep repeating this step, centrifuging at 2500 G till the time you concentrate this soup to around 40x. Okay, and you have to bring them to a volume of around 0.5 ml. It will be easier for you to do a centrifugation, in the next subsequent steps. So here what happens is when you centrifuge them, you'll see that you'll get a flow through. You have to discard this part and you have to collect this concentrated supernatant, transfer them in the clean tube, and you can store this. Again, you can store this in 4 degree if you are going to use it next day, or if you are going to use it say after a week or a month, you need to store them at -80, and you can thaw them and then purify that. So here I'm going to explain the method of purification using sucrose cushion here. So what I prefer to do is a 20% sucrose, in DMEM. But again, this percentage may vary according to the density of your viral particles. You need to know the density of your particular virus. For SARS-CoV-2, 20% sucrose is the best density that allows the particles to settle down here, while leaving the lighter parts, undamaged parts, everything else in the here in the soup. So this is how it goes. You need to first take the now to set up this condition. You need to first put the serum free media, and then you have to take this sucrose and you have to use a syringe to gently gently put the sucrose here beneath this, right? And since the density of the sucrose is more, it will automatically stay settled properly here. And then take the 0.5 ml of your concentrated VLP containing soup and add it on the top of it. Okay, and then you have to again centrifuge at 4 degrees centigrade at 6,500 rpm for 24 hours. I have used SW28 rotor, spring rotor for the Beckman centrifuge, Beckman ultra centrifuge. So after 24 hours, you'll see a very fine pellet here, or it may not be visible depending upon how much particles you have made. Like suppose if you are collecting it from just one tube, you may not see the pellet, but if you pool the 3 or 4 tubes in one, and then when you try to purify, you may see some white pellet here. But whether you see it here or not, consider that it is definitely there. Okay, so gently start sucking the soup from here on top, and start sucking in, get rid of all this part, and just last the pellet will be there, and you need to reconstitute the pellet again or resuspend the pellet in the serum free medium, maybe 100 microliter or 50 microliter, depending upon whether you want them concentrated or little diluted. I prefer to do it in a 100 microliter volume here. So this is the pure VLP that you get in serum free medium now, and which is ready to be used. And hopefully these particles are intact particles, because again I told, when you use a sucrose cushion, that allows you to purify the intact particles only. Only intact particles of a particular density will pass through this density and settle down here. The rest will not be able to pass. So that is how you purify them. Now let's talk about the utilization of these virus-like particles into research. So these particles or the virus-like particles have been in research for several purposes. Let's discuss a few of them briefly here. Utilization in virus research first. So these VLPs can be used to study viral structure, their assembly and invasion mechanisms. By producing VLPs in vitro, the effect of mutations on viral assembly and infection can be studied. Now this can be very useful for understanding virus-host interactions and developing antiviral therapies. Use them in the vaccine development. Now these VLPs, as I told, they closely mimic the structure of viruses. Now since they lack the genetic material to replicate, they serve as an excellent vaccine candidate. These VLPs are capable of eliciting robust immune response without causing disease, and that is why they are good candidates. And some of these VLP-based vaccines have already been developed against viruses and are also FDA approved. Let's take an example of HBV, hepatitis B virus, and we have the vaccine named Engerix B. The targeted epitope is the S antigen of the virus, and the vaccine is developed by GSK in the yeast system. Another example can be the vaccine for the HPV, that is for human papilloma virus, and its name is Gardasil. Now, upon these, VLPs and their success as, VLP vaccines, several VLP-based vaccines for other viruses such as HIV, Ebola virus, SARS-CoV-2, respiratory syncytial virus, and influenza virus are still under different, clinical trial stages. Utilization can be in the drug delivery. Now, these VLPs can be engineered to encapsulate and deliver drugs or genes to specific cells or tissues. Their virus-like properties enable them to be efficiently, enter the cells and making them promising carrier for the targeted drug delivery. Can also be used in the structural biology. Now, these VLPs are excellent tools for structural biology studies, such as like determining high resolution structures using advanced techniques like X-ray crystallography or cryo-electron microscopy. Now, such studies provide insights into the organization and assembly of viral proteins, and thus they help in the development of antiviral drugs and vaccines. Utilization is as a diagnostic tool. Now, these VLPs can be used in diagnostic assays to detect specific antibodies or antigens associated with viral infections. They can serve as antigens to detect antibodies in patient clinical samples or as carriers for antigens in diagnostic tests. Uh, I'll discuss the last, one more last utilization, which can be in the cancer therapy. Now, these VLPs can also help in cancer therapy. For example, they can be engineered to display tumor-specific antigens and thus making them promising candidates for cancer immunotherapy. By stimulating the immune system to recognize and attack cancer cells, these VLP-based vaccines, they hold a great potential for cancer treatment. So overall, I would say that the versatile nature of VLPs make them a valuable tool in a wide range of research fields, and research and development efforts must continue to explore and expand, their applications. So thank you so much for watching the video. If you have any specific questions, feel free to write to me, and I will try to reply to your questions as soon as possible. And if you want me to make videos on any specific topic, just drop your suggestion in the comment box and I'll try to make the next video on that. And if you have gained useful information, channel, and don't forget to hit the bell icon to get the latest updates on new videos. Thanks once again.